welcome everyone. I'm Louis-Pascal Xhonneux, as a very kind introduction. I'm a third-year PhD student under Professor Jian Tang, who unfortunately couldn't make it in person, so I'm here presenting on his behalf. These are his slides. And I'll be presenting the work we've done in our group towards using geometric deep learning and knowledge graph reasoning for drug discovery. So why is drug discovery an interesting target? Well, it takes a very long time and it is very expensive to bring new drugs and treatments to the market. Especially there's this first phase on the left-hand side here, where we are trying to identify possible targets, so proteins or genes to target with drugs. And then these drugs need to be optimized so that they're soluble and non-toxic. And then even at that stage, we have no idea whether they will work. So there's an extensive testing phase, phase 1, phase 2, phase 3. And so trying to find good drugs to test is really important before we start phase 1, phase 2, and phase 3. Drug discovery is an important area, but we also need to make sure that AI is actually applicable in this area. And typically for AI and machine learning, we need a lot of data. And it turns out that the biomedical domain is actually one of the scientific domains that has been producing the most amount of data in recent years. You might have heard about projects like sequencing the human genome. In particular, a lot of these compounds from scientific literature and patents and whatnot have been collected in a very large database called ChemBL, which has like 1,800,000 compounds, so that should be sufficient for any deep learning application. So talking about data, what does our data actually look like? So in our group, we are focusing on 3 main types of data. They're all graph structured based or 3D structures. So quite commonly, we will be working on small molecules because most drug-like molecules will be small molecules of like a dozen to several dozen molecules. We may represent these molecules as graph networks where individual atoms are nodes and covalent bonds are edges. Or sometimes we also have additional information like the 3D structure and we'll have coordinates for each of the atoms. Now, obviously, proteins are very important. They are kind of how our body kind of uses proteins in many places, and they're often targets to try and address diseases and stop infections. And again, proteins can be represented either as a graph, where we'll have residues in the proteins, and the edges will be distances and angles between the residues, or we may have just the 3D structure. The third type is more high level. Here, this is a knowledge graph, so this will contain entities such as diseases, drugs, genes, proteins, and we'll then link them with some sort of relation. So a relation might be, for instance, this drug blocks this protein production. And biomedical knowledge graphs sort of capture the knowledge we already have, but they often allow us to infer more new knowledge. And this is what knowledge graph reasoning will be about. In this talk, I will begin by introducing some of the work we've done on molecule and protein modeling. I'll then talk about the work we've done in knowledge graph reasoning. And finally, I'll briefly introduce our open source library, which has a lot of the models implemented, so practitioners can kind of get started quickly. Initially, a lot of the work done in drug discovery were graph-based methods. And so here are some of the kind of important problems in drug discovery with graph-based methods. The first is, if we have a molecular graph, we may want to predict properties such as solubility, toxicity, charge density. And the problem is that a lot of the time, we won't have this kind of data, and labeled data is expensive. So we want to predict this data. Because often there's not enough labeled data, self-supervised and pre-training are important factors in this. The next problem is kind of the inverse of this. So given that we have some properties we want our molecule to satisfy, can we generate such a molecule? So in this kind of situation, deep generative models that have been kind of applied in other domains, such as images, must be adapted to kind of predict graph structures, which carry a lot of additional challenges, such as permutation invariance. And then these molecules may also then be optimized further, similar to in the drug discovery process, where you'll often also see initial drug finds being optimized. These may then often be optimized with something like reinforcement learning. And finally, if we know a molecule that we want to produce, we still need to know how to synthesize it, because we don't just have a machine that just prints out the molecule we need. And so this is then a problem of, given some reactants, we want to find a synthesis process of chemical reactions that gives us the final molecule, and this is called orthosynthesis. And AI has also been making inroads there. I won't go into too much detail into these works, and instead we'll go straight into the geometric deep learning. So 2D graphs are nice, but a lot of the properties won't be captured in a 2D graph. So molecules obviously exist in 3D, and this 3D structure is critical. So for instance, we often talk about how molecules and proteins are like keys and locks. And this key and lock is kind of like a 3D system. So the 3D conformation of molecules is really important. It tells us also a lot about its properties. Now, something to keep in mind is that any given molecule may have several conformations. And these conformations may change depending on environmental factors such as temperature and pressure, for instance. So in the next stage, we'll be focusing on 3D conformations. So the first problem is that these 3D structures are often unavailable. It's quite expensive to find the 3D structure of a molecule because it requires expensive and time-consuming crystallography experiments. And so kind of the first obvious task for machine learning is, given some of the data we've already collected, can we learn how to generate the 3D conformations given a 2D graph? So this is a generative problem where we have a graph G, and we want to predict the distribution of conformations. Because as I mentioned, every graph will have several conformations. Now, there are some immediate problems that you'll notice. In physics, there are a lot of symmetries. And so, for instance, as you can see, this molecule is rotating, but the 3D conformation, the actual structure, hasn't changed. But the coordinates will. So if you rotate it, the coordinates, absolute coordinates will change, but actually the structure is the same. And so neural networks aren't inherently built for that. They will just give you a number, but they won't respect any symmetries whatsoever. So one of the first problems in this field is, how can we make sure that we satisfy the most basic physical symmetries? I'll present one work in a bit more detail, which is a recent work published at ICLR 2022 by my colleague Minkai Xu. So diffusion models, diffusion processes are a type of generation process. Briefly, the way they work is, given we have a real distribution, C0, that we want to sample from, we create two processes. We create a forward process in which we just add noise to our distribution until it just looks like a noise distribution. We've added so much noise that the original distribution has kind of vanished. This is kind of a fixed process. It's easy enough. What we're interested in is the reverse process. Now, given that we have the forward process, can we learn the other direction of the process? And this is the kind of the generative process, where at some point we can then just sample from a noise distribution and generate the molecule. So we'll be interested in learning this distribution P, C, T minus 1, given the graph and the previous conformation. I apologize for the math. We have the forward process. As you can see, it's simply a Gaussian kernel. And you'll see we have this beta, which is basically the step size. Basically, initially, we want to add little amount of noise, and later on, we want to add a lot of noise to get closer to the noise distribution. In the generative reverse process, we will also have a Gaussian kernel, but now the key parameter to learn is the mean. So the mean basically represents the direction in which you want to move a given atom in this conformation. And the variance is basically your step size. How much do you want to move on average? So yeah, the key parameters are all hidden in learning this mean of the Gaussian kernel, which represents the forward process. So now the question is, how can we ensure that we respect the physical symmetries? So this respecting of the physical symmetries is called equivariance. So basically, if we turn our conformation for putting it in, we also want to turn our output result. This is what equivariance means. Invariance means that whatever we predict doesn't change even if we rotate or move the initial input. SE3 is simply what we call the physical symmetries of translation and rotation in 3D space. So how do we ensure that our final distribution is SE3 invariant? Well, if we start with an invariant density function, like our noise distribution is SE3 invariant, let's just assume we can sample from such a distribution. If we can ensure that our kernel, so our function that determines the mean in the previous slide that is based on the confirmation, this mu theta, if that is equivariant, so if we move the confirmation, we also move the prediction of the mean, then it turns out our final distribution will be SE3 invariant. So the idea is we start with an SE3 invariant distribution, we ensure that our kernel is SE3 covariant, and then we will get an SE3 invariant density. And so the contribution was to make this work and to find a good SE3 covariant kernel. How do we sample from an SE3 invariant density? So there's a relatively neat way to get canonical coordinates in terms of translation. We can simply take the center of mass of the molecule and always ensure that it's at the origin. This is kind of a nice way to define a canonical set of coordinates for the translation. The problem is with rotation, there is no canonical orientation of a molecule. However, if we use a Gaussian distribution is rotation invariant. And so if we move the center of mass to 0 of the molecule and we sample it from a Gaussian distribution, we will have rotation invariance and translation invariance. To ensure that the kernel is SE3 covariant, we have this network epsilon theta. And this network epsilon theta kind of uses the distance between nodes, or between positions. And obviously the distance between atoms won't change if you rotate or translate it. And so this use of invariant inputs from the confirmation then allows the kind of final shift of the position to be equivariant with respect to the confirmation. And this then satisfies our requirement for being an SE3 covariant kernel. Briefly looking at the results, the COV means coverage. So given that we have a set of confirmations we've predicted and a set of true confirmations, we want to see how much of the true confirmations we've covered, so how many of them we found. The R stands for recall, and the P stands for precision. Matching is how close they are on average. As you can see, compared to prior work in bold, the numbers have significantly improved, and we're able to generate much better confirmation. And this is roughly what a confirmation diffusion process would look like. It starts off with a lot of noise and then finds its final position and looks like an actual molecule with a small amount of noise. Now, a different problem is that for molecules, proteins, we often want to find representation. Deep learning is about finding good representations. These representations can then be used in downstream tasks. Now, how can we find good protein representations without a particular task in mind? So prior work has treated proteins, which can be described as a sequence of amino acids, so it's kind of a 1D sequence, as simply a language model. So they've taken language models that work on normal vocabulary and have instead trained them on the vocabulary of proteins. So an amino acid sequence is kind of just treated like a sentence. This had decent results, but in this particular work, they show that if you pre-train on the 3D structures of the proteins we have available, such as using a type of self-supervised learning. So language models are typically trained with, you mask a part of the sentence, you mask like 10 or 15% of your actual sequence and then ask the model to predict these missing amino acids. For 3D structures, that obviously can't quite work, so instead you predict things like the distance between residues, the angles, the dihedrals, the residue type, and this kind of self-supervised learning significantly outperforms sequence-based models. And these representations can then be used for downstream tasks, which is very useful. Okay, moving on to biomedical knowledge graph reasoning. So biomedical knowledge graph is highly heterogeneous. So different nodes will mean very different things. Diseases, drugs, proteins, genes are very different things. There's no good similarity between them. But what we really care about is the relation and links between these. So this has several applications. Drug repurposing is one of them. So given that we have an existing drug that we know relates to an existing disease, we may want to infer if there are any other diseases it may link to. This is actually quite common. I think drug repurposing is actually the majority of treatments being submitted to the FDA is simply drugs being repurposed. The other part is target identification, which is often the first step in the drug discovery process. This is where we want to predict the links between, for instance, between a disease and genes and proteins. So these genes and proteins can then be the target of our drug-like molecule. Now, fundamentally, this is a problem where we have a graph and we want to predict new links in this graph. So mathematically, we formulate this as we have some head entity. This might be the disease. We have a relation Q. This might be, links to a drug or something. And then we want to find a target entity V, given our current knowledge base. So we have had some recent work on knowledge graphs called neural Bellman-Ford networks. And the idea of neural Bellman-Ford networks was to combine the benefits of path-based methods with neural networks. So traditional path-based methods include something like personalized PageRank, which you might have heard about. It's how Google search results work. Graph distance, which is simply the distance between two nodes in a graph. These path-based methods have various benefits. They're often interpretable because we have an actual path, which can kind of represent the reasoning path, and so can kind of be verified. They're inductive. They don't have like a representation of the entities that ties them to this particular knowledge graph. They can just be reused on any particular graph. And if you're clever about it, you can make them scalable with dynamic programming. The problem is that they have very limited capacity, so they're not really learning much, and thus the generalization ability is kind of poor. Like they might work on one graph, but it's unlikely that they'll work on another graph. So the question is, can I parameterize these with neural networks? So to better understand what we did, it's worth talking a bit more in detail about specific path-based methods. So for instance, PageRank really considers a set of paths between two nodes, all the set of paths, and each path is weighted according to the product of the probabilities on each edge. So if we have a probability 1/3 on an edge, we multiply this by we multiply all these probabilities together. And then to kind of get a pair representation between this blue and orange node, we'll sum all these paths together. Now, a different example is the graph distance, where instead of multiplying the edges, which we interpreted as edge weights that we interpreted as probabilities, we now simply take them as the distance and add them together. And instead of adding all the paths together, we take the minimum. Now, what all of these path-based methods have in common is that they can be understood as a semi-ring. So a semi-ring basically says that we have two binary operations, and you have some basic laws like associativity that hold for these two relations. But basically, most path-based methods can be all described as one of these semi-rings. So our point was, what if instead of picking a particular multiplication and summation operator, we can just learn them instead? And this way, we're learning a path-based method that should hopefully be generalizable and has kind of a capacity. So these summation and products will be neurally instantiated. This has various benefits. We will use a dynamic programming approach, so they will be scalable. We keep the inductivity and the interpretability because we're still just doing a path-based method. And now, rather than being a handcrafted, we'll have a neural parameterization, which is really what we want to do. So I kind of explained this, but in kind of maths on a page, the representation between two nodes U and V, given some query, will basically be the sum of all the paths, and the paths are represented by the product of all the weights along the edges, where now the sum and the product are arbitrary binary operators that we define and learn a priori. So we use the generalized Bellman-Ford algorithm to actually compute this representation. So if you don't know, the Bellman-Ford algorithm is the shortest path distance computation algorithm that is a dynamic programming method. It basically is a particular way to compute all of these paths. The number of paths will be exponential in the number of nodes in such a way that you can avoid the exponential explosion. Yeah, so this is the particular method. Now, kind of visualizing what the sum and what the product operator is, graph neural networks have this aggregate message type framework, and so we will have an indicator network to kind of learn the initial representation, so the indicator will be 1 if U is equal to V, Otherwise, I'll put a representation for 0 if u is not equal to v. And then we can start generating the representation for all the paths between two nodes that are of length t. And so as we continue, as we do more and more iterations, we'll consider more and longer paths. But as you can see, the message function is a neural network, so we've learned kind of a binary multiplication operator. So yeah, the embedding is learnable and depends on the relation, so on the question we're asking. The message function simply represents on the weight as well as the current representation. The aggregation will be some form of permutation invariant function, sum, mean, max, PNA. But the point of this is that at no point did we have a fixed entity embedding that we used. So since the only thing for which we actually have an embedding are the relations, this is inductive. So we can just pre-train our method on one graph, on one biological knowledge graph, and then apply it to another. Another important point of this is that they're very parameter efficient. So the current state-of-the-art methods for knowledge graphs are like embedding-based methods. And you can see, since they need an embedding for each node, that takes like 45 gigabytes for a given data set, while we only need relations for, like embeddings for the relations, and so we just have 60 megabytes. Besides all the benefits of being inductive, scalable, it also just is much superior to the previous embedding methods and vastly outperforms them, which is very nice. Okay, finally getting to TorchDrug. TorchDrug is our open source machine learning framework that we've kind of developed in the group to kind of make things easier for us. It'll contain a lot of baseline models that we need to use to reproduce results. We're trying to add our own methods as we develop them to this as well. And it kind of contains a lot of boilerplate code we might otherwise use. Yeah, if you want to play with any of these methods, check if they've been added to TorchDrug already. We cared about the drug discovery task. We kind of briefly alluded to why it was important and covered a variety of tasks in this talk. We talked about property prediction, which was predicting the properties of molecules. We talked about pre-training representations so that we could use them for downstream tasks, like a general representation that is useful, that contains the information of the molecule. We talked about de novo molecule design, where we might generate molecules to satisfy certain properties, and then we can further optimize these with reinforcement learning. We talked about synthesis, which is how do we synthesize the molecule. We talked about biomedical knowledge graph reasoning, and we talked about protein representation learning. And these are kind of some of the main focuses of our group in terms of drug discovery. Yeah, so the takeaway is there's lots of data, which is always good for deep learning. It's quite an important application area. There's field-specific challenges, but that's what makes it interesting from a research point of view. And we've covered graph-based methods, geometric deep learning methods, and knowledge graph methods. Thank you very much.
Do you think that these methods can be used for new materials discovery, like biomaterials and polymers?
So materials are actually even trickier than just drug-like molecules to present. They have additional challenges. But yes, work has been happening on this. People have tried to do crystal prediction, where given a molecule, we try to predict the crystal, because any pill you take really contains crystals. And so this is an important problem. And material discovery is incredibly difficult. So there has been work on this, but not specifically in our group just yet.
Thank you. It's a great talk. I'm wondering, you present different aspects of your graph representation for different tasks, like the 3D structure, like the knowledge graph. But did you try to combine all together, for example, to see what, if you take a molecule that you know have a specific interaction from your base knowledge graph and apply that to the structure? For example, if you try to think about how some, interaction with other molecule, or in the case of a specific disease, if this has an impact on the 3D structure of the molecules and so on.
So this is usually broken down to subproblems. So for instance, one particular subproblem I know Andrea Diak has worked on is drug-drug interaction, whether two drugs, if used together, have a synergistic or toxic effect. So that's, I guess, the type of problem you're referring to. Another problem that people have been very interested in is, given some molecule and a protein, can we know where the where the drug-like molecule will fit into this protein. So it's usually formulated as a new task and usually tends to require some new methods. So it's, yeah.
Is it already possible to optimize simultaneously for the drug effect, a given effect on human body or on an illness, for instance, and also on the easiness of synthesizing the molecule?
So the first question, like, to what extent can we optimize molecules? So typically, the optimization of molecules happens with respect to specific properties rather than a disease, because that is much more complicated and involves also the protein that you're targeting within that, like within the kind of replication chain of the bacteria, for instance. But yes, there have been optimizations, for instance, to increase solubility. You sometimes run into problems where the machine learning model learns to do something ridiculous, which isn't particularly useful. But yes, this has been done. Your second question was?
Yeah, can you optimize for easiness of synthesizing the drug after that?
So I don't think people have really tried that, because most people, like currently the most important problem is, can we just make the drug less toxic and stuff? And usually we are able to find a synthesis path. I'm sure that industry will be very interested in actually finding a cheaper way. I think cheaper is really what the metric they'll care about to produce this drug. But I think pharmaceutical companies will figure that out.
Merci beaucoup pour la conférence. Je pense que vous aviez tous des super questions. Je vais essayer de faire un résumé rapide, puis ensuite on va pouvoir aller à la pause dîner. Donc, on se demandait si l'apprentissage par géométrique et 3D pouvait nous aider dans la découverte des médicaments. La réponse est quand même très clairement oui. Après ça, jusqu'à quel point, c'est sûr qu'il y a des choses plus complexes qui s'en viennent, mais je pense que la réponse est clairement oui. Donc, on sait que la découverte des médicaments, c'est un processus qui est très long et qui est coûteux, puis qu'on a besoin de beaucoup de données. Heureusement, comme on a mentionné, il y a beaucoup de données en biomédical. Puis, c'est un des domaines, en fait, où est-ce qu'on produit le plus de données, et c'est là-dessus qu'on va s'appuyer pour créer nos modèles 3D et nos graphes. Donc, on cherche à trouver, premièrement, comment une molécule réagit et aussi, parallèlement, comment créer une molécule pour qu'elle réagisse d'une certaine manière. Donc, on pose des questions un peu des deux côtés quand on s'intéresse à comment les molécules sont faites. Puis, finalement, la troisième question et la dernière, ça serait comment synthétiser cette molécule-là qu'on veut produire. Donc, pour y arriver, on veut représenter les molécules en 3D pour augmenter notre compréhension. Étant donné qu'en ce moment, on les représente surtout en 2D, mais on sait qu'en 3D, ça serait beaucoup plus utile. Par contre, la majorité des modèles 3D ne sont pas encore disponibles. Donc, ce que je crois avoir compris que vous avez fait, en fait, vous avez premièrement fait un procédé direct, donc qui était de voir la molécule, d'ajouter du bruit pour ensuite essayer de faire le procédé inverse et donc d'enlever le bruit pour voir la vraie molécule tout en respectant la symétrie. Donc, ça, c'était pour comment modéliser les molécules en 3D, qui est la première partie de votre projet. Puis ensuite, ce serait de faire les graphes, donc de pouvoir prédire les maladies qui seraient liées aux médicaments et les liens entre les maladies et les gènes et les protéines. Puis, avec tous les calculs, on a vu que ça s'en venait quand même possible de prédire ces liens. Parce que, en fait, ce qu'on voulait, nous, c'était pas de les faire à la main, les liens, comme ça se fait en ce moment, mais on veut que la machine soit capable de l'apprendre par elle-même. Donc, que ça devienne un modèle qui est plus inductif et évolutif. Finalement, je vous laisserai sur le Touch Drug, qui, je pense, est un bon outil à utiliser pour tous. Et sinon, pour ouvrir la discussion encore demain vers 10h45, je pense qu'il y a la Patricia Conrad qui va parler de est-ce que ça va être possible d'utiliser la médecine préventive, d'utiliser l'intelligence artificielle pour la médecine préventive. Est-ce que ça va être possible ou on n'aura pas le droit? Donc, ça va poursuivre un peu la discussion ici. Je vous souhaite un bon dîner, puis on se retrouve cet après-midi.